Good morning, everybody. Today I'm here to present an antiproliferative activity of Phyllanthus debilis Klein soil plant. Healing with medicinal plants is an all as mankind itself. So the connection between man and his search for drugs in nature dates from the far past, of which there is ample of evidences of various sources plants are using as medicine. Natural products remain an important source of new drugs. The plant-based drug discovery resulted mainly the development of anti-cancer agents like vincristine, vinblastine, etoposide, placitaxel, camptothecin, etc. Sri Lanka is a pool of natural herbs, and Sri Lankan traditional medicine and combination of those two will lead to novel drug development studies. So from that Sri Lankan natural pool, we selected Phyllanthus debilis Klein soil plant in Sinhala known as Elapitavakka. So this plant is using in traditional medicine in Sri Lanka to treat hepatitis and jaundice. Phyllanthus debilis plant belongs to genus Phyllanthus, family Phylanthaceae in the major group angiosperms or flowering plants. Genus Phyllanthus consists of more than 1,000 species, of which many are used as traditional medicines, used for treating hypertension, diabetes, hepatic, urinary and sexual disorders, and other common ailments. Phyllanthus debilis is an annual herb. It is common weed in waste and cultivated grounds in Sri Lanka. So phytochemicals are responsible for all the biological activities. Phyto means plants in Greek. Phytochemicals are non-nutritive plant chemicals that have protective or disease-preventive properties. Plant produces these chemicals to protect themselves, but they can also protect humans against diseases. But they're not required by the human body to sustain life. So the objective of this study was to investigate antioxidant and antiproliferative activity of water extract of the Phyllanthus debilis plant. So in the methodology, plant material was collected from Panipiti area, Colombo district, 2014 June, and authenticated by at Bandaranaike Memorial Ayurveda Research Institute. Then washed with distilled water and deionized water and dried. Powdered sample was mixed with deionized water and refluxed and filtered. That filter was centrifuged, and decoctions were filtered using filter papers. Then supernatant was freeze-dried, weighed and stored in -20 in sterile glass bottles until further use. So in antioxidant activity, DPPH assay was used. DPPH is a stable free radical. Have ability to capture free radicals is due to delocalization of the unpaired electron all over the molecule. So DPPH reagent is in violet color, then it will become into yellow colored with the antioxidants present in the herbal extracts. So different concentrations of the plant extract and ascorbic acid positive control mixed with the DPPH reagent kept in dark for 30 minutes. Then the absorbance values were taken at 417 nanometers. Results were expressed as a percentage inhibition as given in the formulae. In the cell culture studies, rhabdomyosarcoma cell line was used, and the DMEM medium with 10% FBS and the glutamine, penicillin, streptomycin and heaps, 5% CO2 incubator, 37 degrees at humidified environment. Cells were grown in 24 well culture plates and allowed to attach overnight. Negative controls and positive control cyclohexamide was used. 24, 48, 72 hours experiments was done for the carried out for the cell morphology and MTT assays, and the ethidium bromide acridine orange dual staining was carried out for the 24-hour experiments. So the antiproliferative activity, MTT cell viability assay was carried out. So MTT is a yellow colored compound that will convert into violet color by this mitochondrial dehydrogenases present in the live cells in the medium cell environment. So the quantity of the formosome crystals is directly proportional to the number of viable cells. So the media we treated replaced with MTT and incubation, and the HCl isopropyl alcohol was used to dissolve MTT crystals, and absorbance values were taken at 570 nanometers, and the percentage cell viability was calculated. So in the ethidium bromide and acridine orange dual staining, acridine orange is a cell-permeable dye that intercalates DNA and RNA, but ethidium bromide is not cell-permeable, so it can only stain cells that had lost an intact cell membrane. So ethidium bromide and acridine orange dye mixture was added to each well after removing the media, and cell morphology was observed and recorded. So according to NTT cell viability assay, EC50 values are as given in the figure. So it contains very high antioxidant, with the time period it's reducing its cell viability. The initially 24-hour incubation period to 87.2, and then it will come to 67 at the 72-hour exposure. With that, visual observations are also helping for this thing. When it comes to microscopic observations, negative control, so confluent monolayer can be observed, but with the positive control and when increase in drug concentration, so positive control like results can be could be observed for the 24-hour exposure. So the shrinked and rounded cells like in positive control, and blebbings also can be seen in higher concentrations. So in ethidium bromide acridine orange staining, live cells are in green color. Normally early apoptotic cells are in yellow color. Late apoptotic are in orange, and necrotic cells are in orange to red. So this is positive control with cyclohexamide. When increase in drug concentration, apoptotic cells and the late apoptotic cells can be observed like in the figure. So when it comes to antioxidant values, it has a higher antioxidant capacity. It's according to the results, 3.26 ascorbic acid EC50 value and the 5.89 EC50 value for the DPPH free radical scavenging assay. So the antioxidant values are also higher for the plant extract. So conclusion was Phyllanthus debilis plant water extract has a high antioxidant activity and moderate antiproliferative activity, and showing apoptotic or programmed cell death characteristics at high concentrations qualitatively. These are my references, and I would like to thank my colleagues and department staffs. Thank you.
Thank you, and it is open for discussion.
So this DPPA, radical quenching experiments, with plant extracts, generally can lead to anyway, some quenching results, that you, might mislead to say that, it has a very high antioxidant properties and all. So again, what concentration you have used these extracts, might play a major role, while quenching the radical in the molecule, right? So any, basically radical quenching can be done by any, oxidant that you can have in a plant extract, right? So basically, have you done a control experiment, maybe taking some other leaves extract and then compare that to the one extract that you basically obtained from your medicinal plant?
It was compared with the ascorbic acid.
But that's a chemical, right? So have you done with the plant extract to extract?
Yes, with another extract. extracts also I have carried out.
But you haven't shown that data here, right?
DP page as a result. So that slide was corrupted.
It's not for you, it's for everyone. I'm extremely impressed from wonderful presentation with so much of enthusiasm, and one advice is that regardless of what you do, the most important thing which you eventually will achieve is in vivo activity. Nobody on the face of the earth will be interested in your work unless you demonstrate that your formulation, your compound is actually working. It is extremely important that Sri Lanka or other countries should develop national animal science laboratory facilities with disease model, and then you demonstrate that your formulation, your plant, your compound is actually working in vivo. It doesn't really make much of a difference whether you know the mechanism of action or not. Once you demonstrate that this particular formulation function in vivo, then you would be able to attract lots of international attention, especially pharmaceutical companies, and they would do rest of your work. Do not worry, but you can only attract attention of the world when you demonstrate that your formulation is actually working in fully integrated animal model.
In your MTTC assay, what was the concentration used for positive control?
50 micrograms, 5 micrograms per ml.
Thank you.